All right, Chemistry 102. This is practice exam 5. Question number 1. Which of the following statements concerning enzymes is false? We have inhibitors are molecules that cause an enzyme to lose activity. That's definitely true. Enzymes are the body's biological catalyst. Well, that's the definition of an enzyme, and they increase the rate of cellular reactions. That's also true. Enzymes are usually globular proteins with a flexible three-dimensional shape. Yes, because we know that the conformation of an enzyme can change, or another way we put it is that it's flexible. It can move to some degree. Next one, D, the reactant in an enzyme catalyzed reaction is called a cofactor. That is in fact false, right? We are looking for a false statement. This is false. It is not called a cofactor. It is called a substrate. So that's the correct answer. The last one, E, is the active site of an enzyme is where the substrate binding and catalysis occur, and that's true as well. Question number 2, sucrase is an example of an enzyme that displays absolute specificity. What does the term absolute specificity indicate about an enzyme? Well, when I hear the term absolute specificity, to me, it means that it catalyzes a reaction for one specific molecule. Okay, so one specific molecule, which means that it's one specific substrate, right? One specific starting material, if you will. So A, it catalyzes the reaction of only one class of compounds. No, that would be group specificity. So we'll cross that out. That would be group specificity. It catalyzes the reaction of only a single type of bond. No, that would be linkage specificity. It catalyzes the reaction of only a single substrate. That is our correct answer. D, it catalyzes the reaction of only a single enantiomer of a compound. No, that would be stereochemical specificity, stereochemical. And it catalyzes one type of reaction for the same class of compounds. No, not that either. So absolute specificity means only one specific type of substrate, a single substrate. Question 3, which of the following describes how an enzyme can affect the transition state in an enzyme catalyzed reaction? We spent a lot of time looking at the enzyme substrate complex, how you get to the activated complex, and how you end up forming the product, and the product gets released. Well, let's take a look. One of the ways that an enzyme can affect the transition state in an enzyme catalyzed reaction is by placing a stress on a bond. That is definitely true. We've seen an example of that in our class, bringing two reactants together in close proximity and in a suitable orientation. A good example of that would be something like sucrose synthase, which we looked at. Modifying the pH of the environment, yes. None of the choices are correct. Well, that's definitely not true. In fact, all of the choices, A, B, and C, are all correct. And so the answer is E. Question number 4, feedback inhibition. In feedback inhibition of an enzyme catalyzed biosynthetic pathway, what frequently serves as a negative allosteric inhibitor? Right? If you think about, a negative allosteric effector. If you think about the example that they used in the book, they said negative allosterism is kind of like a thermostat, right? You take a thermostat in a room and you turn up the heat, but as the heat rises in the room, as the temperature rises in the room, what happens is once you attain the desired temperature, it shuts off the thermostat, doesn't it? It triggers an effect that shuts off the thermostat. And so if we look at the possible answers here, we have oxygen, the final product of the pathway, the starting material, the temperature, acetylcholine. Well, the answer is the final product of the pathway, right? We saw several examples how we had an enzymatic pathway where we had maybe, I don't know, a series of 1 or 2, or when I say a series, I should start with 2, right? 2 or 3 or 4 or 5 steps, doesn't matter how many, but we saw how sometimes the product can inhibit an enzyme at an earlier pathway, thus being a negative allosteric effector, right? It can react or bind to another enzyme in the pathway and it shuts down the pathway. So the final product of the pathway can make a good negative allosteric effector. Question 5, which of the following describes pepsinogen? Pepsinogen is a zymogen. Well, we went over that in class, so a zymogen. Zymogen. Okay. Or a proenzyme, same thing, pro. So pepsinogen, it is it is a cofactor for the enzyme pepsin. It is the inactive form of the enzyme pepsin. It is the inhibitor of the enzyme pepsin. It is the name of the reaction catalyzed by pepsin, or it is an isoenzyme of pepsin. Does anybody have an answer for this one? Which one best describes pepsinogen? And if you know the definition of a zymogen or a proenzyme, you should be able to get this one pretty quickly. Yeah, thanks, Peyton. It's the inactive form of pepsin, right? If you think about chymotrypsinogen or pepsinogen, those, whenever you see inogen, that's a zymogen, and it's just the inactive form of an enzyme, and then it has to be activated in some way. So again, whenever you see inogen, it means inactive form, okay? Inactive form of enzyme. All right. Question number 6. Proteases or proteolytic enzymes are responsible for which of the following functions? Well, when I see the word proteases, what comes to my mind would be a protein, right? So what kind of bonds are found in proteins? Well, we know that proteins are all held together with amide bonds. If I just draw a generic amide bond like this, okay? A protease is going to be responsible for hydrolyzing peptide bonds between amino acids and proteins. That's the correct answer. Saw it right away. Okay, none of these other answers are even remotely close. Right? You should know what a protease is. Right? You should know that a protease is going to hydrolyze a peptide bond between amino acids. You should know what a peptide bond is. Right? A peptide bond is simply an amide bond that is found in a protein. All right, well, let's take a look at question number 7, which shows us a picture of a protein, right? We have our NCC, 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 so that looks like a pretty good tripeptide. It says that the protease enzyme chymotrypsin cleaves peptide bonds on the carbonyl side of aromatic amino acids. Which labeled bond in the peptide below would be cleaved by chymotrypsin? Can anybody tell me which one of these bonds would be the one that would be cleaved? Would it be A, B, C, D, or E? On the carbonyl side of an aromatic amino acid. Okay, let's take a look at it. So let's analyze this tripeptide. Now, if you're like, Mr. Dion, how did you know it's a tripeptide? Remember that we have NCC, that would represent one amino acid, right? NCC, so on and so forth. So if I look at my molecule that's here, I'll use my green pen. We have nitrogen, carbon, carbonyl, so that's one amino acid, or I could box in the whole thing. So there's one amino acid. Let me use the yellow pen here. I have NCC here. This is my second amino acid. And then I have NCC, and this is my last amino acid. Which one of these is an aromatic amino acid? Would it be the one in green, yellow, or red? And it's not a trick question. Yes, it's the one in yellow, right? Because this is an aromatic ring. Well, if it goes nitrogen, carbon, carbonyl, we're looking for the carbonyl side, right? It says it cleaves the peptide bond on the carbonyl side of the aromatic amino acid. Here's the carbonyl on the aromatic amino acid, and here's the peptide bond, right? This here is a peptide bond between the carbonyl and the nitrogen. So the bond that would be broken would be E. That is the bond in question, okay? Remember, a peptide bond is the bond between the carbonyl carbon and the nitrogen. All right, I know that you can all recognize an amide when you see it, but the peptide bond is this bond right here, the one between the carbon and the nitrogen. How many peptide bonds are in this molecule, in this tripeptide? There's only two, right? I'll use my blue highlighter. This is a peptide bond, and this is a peptide bond. Those are the only two peptide bonds in the entire molecule. But E is on the carbonyl side of the aromatic amino acid. Give me a thumbs up if that makes sense, or stop me if you have a question about that one. I want my students to be able to recognize structure and recognize functional groups and bonds. Okay, cool. All right. Well, again, stop me if you have a question about those kinds of things, because I consider those very, very important. Hey, could anybody name the amino acids that are in here? You know, I don't expect you to be able to, but anyhow, you can maybe take a look at them and see if you can, you know, memorize them. For my students who might be interested in going into biochemistry or something like that, you can see if you could come up with the names of those. Anyhow, and I'm not going to ask you that on the exam. You don't have to memorize the structure of any amino acids. Anyhow, question number 8. Which of the following is a medical use of enzymes? There's a short section in our textbooks which deals with medical uses of enzymes. Enzyme replacement therapy for missing or malfunctioning enzymes, absolutely. Yes. An enzyme to diagnose damaged tissue or disease in a particular organ, absolutely. Analytical reagents, yes. Injection of enzymes to counteract blood clots, absolutely. And so all of these choices are correct. Enzymes have many uses in medicine. Question 9, what's the effect of an enzyme on the equilibrium constant for a reaction that it catalyzes? Well, if you remember back in Chemistry 101, you would have discussed K equilibrium or equilibrium constant. And we know that the enzyme doesn't have any effect on equilibrium constant, because an enzyme, where is it here? Enzyme increases, we'll put here, increases reaction rate, right? It speeds up the reaction, but does not affect equilibrium, but does not affect equilibrium. Right, the relative energies of the starting material and the product are not changed. And so an enzyme is not going to have any effect on equilibrium whatsoever. It's going to affect the reaction rate, okay? It can change the speed of the reaction, but is not going to affect the concentration of the reactants, their equilibrium concentrations. All right, question 10. To which class of enzymes would you assign an enzyme that carries out the dehydrogenation of a substrate? Well, we've already taken a look at these. Oxidoreductases, those are used in redox reactions, so oxidation reduction reactions. Lyases, what I told you is that whenever I see a lyase, I always think it's got something to do with a double bond, okay? Double bond, like, what's a good lyase? Like lactate dehydrogenase is an example of a lyase. Then we have hydrolase, which is responsible for hydrolysis. What else? We have a transferase. Those are used to transfer groups. So transfer groups, like it could be a phosphoryl group, or maybe an amino group, right? That's another example. Anyhow, so which one of these would be responsible for dehydrogenation? Well, it's going to be an oxidoreductase. Why? Because what is the process of dehydrogenation, right? We're losing hydrogens. And losing hydrogens is going to be an oxidation reaction, right? We know that in organic chemistry, that oxidation is gaining oxygen or losing hydrogen. So if I'm losing hydrogens through a dehydrogenation, I'm going to be doing an oxidation reaction, and so it would be an oxidoreductase. Question 11, for enzyme catalyzed reactions, what happens as the concentration of substrate is increased? So we went over this quite a bit. I'm just going to review it with you here. We looked at a plot of the rate of a reaction versus the concentration of substrate, okay? And we said, well, initially, as you increase the concentration of substrate, the rate will increase relatively linearly, but then it's going to drop off and sort of plateau out like this, okay? Now, the reason why it does that is because the rate is going to increase until it reaches that maximum constant value. Again, why does that happen? The reason that it happens is because you have a finite number of enzymes and thus a finite number of binding pockets, and so, or binding sites, you could call it. And so, even if you keep increasing the concentration of the substrate, you're limited by the number of binding sites that you have. So once you've run out, well, the rate is just going to level off because it reaches that maximum value. Question 12, what portion of an enzyme is in direct contact with the substrate? Well, all the magic occurs at the active site. Hopefully you would nail this one. This is a question I would expect my students to do exceedingly well on. It's none of these here. We never even talked about anything remotely like those, right? The active site is where the substrate binds, held there by London forces or hydrophobic interactions, whatever you want to call it, by things like electrostatic interactions, right? Hydrogen bonding, that's what holds the substrate into the active site. Question 13, an enzyme catalyzes the removal of an amino group from one substrate and its addition to another compound. To what class of enzymes does it belong? An oxidative reductase, kinase, hydrolase, lyase, or a transferase? Can anybody answer this one for me? What would be the best answer here? For 13, what would be the best answer? Yeah, thanks, Sadie. Absolutely. It would be a transferase, right? Because I'm transferring the amino group from one substrate to another compound. And we talked about that just a few seconds ago in this question here when we discussed what a transferase was. It could transfer things like a phosphoryl group or an amino group. And so if we're transferring that amino group, we're dealing with a transferase. It's a question that takes up a lot of space here. Phenylketonuria is an inherited disease caused by the inability to properly to produce a properly working form of the enzyme phenylalanine hydroxylase. This enzyme catalyzes the conversion of the amino acid phenylalanine to the amino acid tyrosine by adding a hydroxyl to the para position of the ring. Based on this information, what is the structure of tyrosine? And so you need to know how to be able to figure out the structure of tyrosine. Now, I told you, you don't have to have the structure of the naturally occurring amino acids memorized. However, you know that an amino acid should have a carboxyl group, right? It should have an alpha carbon, and it should have a nitrogen, right? An amino group. So I'm going to put this like that. So we've got that much. Now, I know that normally I would draw the amino acid like this, okay, with the alpha carbon and then the nitrogen coming up like this. These are the exact same thing. I just drew the nitrogen pointing down because if you look at the possibilities, they all have the nitrogen pointing down like that. Now, what other clue do they give us here? They tell us that this enzyme catalyzes the conversion of phenylalanine to the amino acid tyrosine by adding a hydroxyl group in the para position of the ring. Well, you can see that all of these possible answers have an aromatic ring. So if I'm the para position of an aromatic ring, right, you should know that when you have an aromatic ring, such as this, okay, if you have two substituents that are right next to each other, we call that ortho substitution, right? If you have a substituent here and a substituent here, we call this ortho substitution. If you have a substituent here and another one that's a carbon away from it, like this, we call this meta substitution. And if you have a substituent here and one like this, we call this 1,4 substitution or para substitution. So we are looking for a ring that has a hydroxyl group para to the amino or the amino acid portion. And so C would be the only possible answer, right? It's the only answer where you have all of this spinach and the hydroxyl group that are oriented para to each other. And so the answer has to be C. Give me a thumbs up if you follow me on that one. Again, you don't have to have the structure of tyrosine memorized to solve the problem. You can just figure it out by the clues that are given here. And you have to borrow some of your knowledge from aromatic compounds when we discussed disubstituted aromatic compounds, whether they're ortho, meta, or para. So make sure you're aware of that stepping into the next exam and, of course, your final exam as well. All right. Did I circle the right one? Is A, B? Yeah, I circled the right one. Okay, let's move on from there. I don't know why. Anyhow, I'll just scratch that. Not worry about it. Question number 15. Of the following types of molecular interactions, which is not likely to be involved in the binding of an enzyme to a substrate? We have van der Waals interactions, hydrogen bonds, polar-polar interactions. So that's the same thing as a dipole interaction. Covalent linkages or ionic attractions. Could anybody tell me which one of these is probably not going to be involved in the binding of an enzyme to a substrate? Well, let's go through them, okay? Van der Waals interactions are the same thing as London forces, right? London forces or hydrophobic interactions, that's the same thing. And if we have two nonpolar groups, they're going to be attracted to each other through van der Waals interactions. What about hydrogen bonds? Absolutely, right? If I have a hydroxyl group, let's say, and let's say I have, an amino group, right? We can have a hydrogen bond between the hydrogen and the lone pair on the nitrogen. So yes, polar-polar interaction, that's the same thing as dipole-dipole interactions. Ionic interactions, that would be like a salt bridge. Like let's say you had an ammonium ion that could be attracted to a carboxylate like this. Anyhow, I can draw it better. We have a carboxylate, maybe like this, right? We have an attraction between the positively charged cation and the negatively charged anion. But to have a covalent bond, right? Covalent bonds are very, very strong, right? They're much stronger than any other of these intermolecular forces here, okay? When we think of covalent bonds, that's when you would have something called an irreversible binder, like maybe a poison, okay, that would bond or bind to a substrate and it wouldn't let go. So covalent bonds are not likely to be involved in the binding of an enzyme to a substrate. All right, question 16 says, what are the two ways in which energy of a food source can be released in a catabolic pathway? So this is kind of the kickoff to chapter 21, where we looked at carbohydrate metabolism. So how do we release the energy from a food source in a catabolic pathway? Well, I went over this at the very beginning of chapter 21 with you, and I said you can think about the human body as kind of being a big combustion chamber, right? What do we release? We end up forming ATP from the food that we eat, but we also generate heat, right? And so the answer is we form chemical bond energy in ATP, and we also produce heat, right? That's kind of what the human body is, just a big ATP maker and keep ourselves warm at the same time. All right, question 17. The abbreviated form of the equation for breaking the phosphoanhydride bond of ATP is shown below. What is the missing product of this reaction? Well, if we have ATP, that represents adenosine triphosphate. We form adenosine 5-phosphate diphosphate. We also release energy. Well, what else do we form in this reaction? The question mark is an inorganic phosphate group, right? And we looked at the two-step process for making G6P, right? We saw how the first step was the breaking of ATP or the hydrolysis of ATP to release energy in the form of 7 kilocalories per mole. We produce the inorganic phosphate, which binds to G6P. It costs 3 or 4 kilocalories per mole, and we're left over with energy in the end. So the answer is an inorganic phosphate group. Question 18. What is the primary function of catabolic pathways? The primary function of catabolic pathways is the generation of a particular substance. What is the substance? Who could tell me? What is the whole point behind catabolic pathways? And it's not a trick question. In a catabolic pathway. So catabolic is breaking something down. So we would ingest glucose, right? We take glucose in our diet, and then we break that glucose down and we make ATP. Exactly. So we make ATP. That's catabolism, is breaking down, okay? Breaking down molecules like fats, right? Like proteins and amino acids, and breaking down carbohydrates. All right, digestions of carbohydrates begins in the mouth with the aid of what enzyme? The answer is salivary amylase, and so amylase is the best answer here. So salivary amylase. We start breaking down starches in our mouth the second they hit our saliva. Question 20. What term describes the synthesis of glucose from non-carbohydrate sources? Could anybody tell me what the answer to this one is? Out of all these crazy words that look like they're all related to one another, they all start with the letter G. Yeah, exactly. Thanks, Peyton. It's gluconeogenesis, right? We have gluco, right, for glucose. Then we have synthesis, so that's the neo or the new making. So gluconeogenesis. Let me see here. Here we go. So neogenesis, okay? Synthesis of new and gluco for glucose. And so the answer is gluconeogenesis. 21, what hormone is released from the pancreas in response to low blood sugar levels? Can anybody tell me what hormone is released from the pancreas in response to having low blood sugar? You don't have enough blood sugar, you need to break down glycogen. So the answer would be glucagon, right? You need glucagon to break down glycogen. All right, insulin would be for the other way around, right? I had a friend that I used to go to Five Guys Burgers and Fries up in Boulder with, and he had an insulin pump, and before we'd go in there, he would, increase his insulin intake. And the reason why is because he was going to eat a meal that was full of sugar. So instead of becoming hyperglycemic, he gave himself more insulin, and that was a response to high blood sugar levels. All right, so low blood sugar, your body releases glucagon to break down glycogen and provide you with enough glucose. 22, what polymer is the principal storage form of glucose? Well, we already covered this in the last question. It's glycogen. This is where we store glucose in our muscles and in the liver. And we talked about what the structure of glycogen is. We have alpha linked or alpha 1,4 linked glucose molecules. They have 1,6 branches, and the branches are frequent and short. And we covered the difference between glycogen and amylopectin. What else? Question 23, it says, which of the following statements concerning the digestion of food groups is false? We have A, proteins are broken down into amino acids. Well, there's nothing wrong with that. Digestion of all food groups begins in the mouth. That is definitely false, right? If you think about lipids, fats don't begin to be digested until they get to the duodenum, when they start to get emulsified, and then they start to get broken down in the small intestine. If we look at the other answers, carbohydrates are broken down into glucose and other monosaccharides. That's true. Polysaccharides are broken down into monosaccharides. Correct. Fats are broken down into glycerol and fatty acids. That is also perfectly correct. Question 24. I went over the pentose phosphate pathway really quickly with you, the PPP. And I told you there's not a whole lot that you need to know about that, except for the pentose phosphate pathway is just another pathway for glucose oxidation, and it provides some important molecules like NADPH, which is required for cholesterol synthesis and some other interesting sugar phosphates. But that is really all you need to know about the pentose phosphate pathway. It's an alternate pathway for the metabolism of glucose, and it provides you with NADPH, which again is used in cholesterol synthesis. The answer is B. Question 25, what substrate is converted into G6P by the enzyme hexokinase? Well, this is the first step in glycolysis where we take glucose, right, and we combine it with ATP. The enzyme that's used is hexokinase, and I told you you should know this step. We end up making G6P, which is glucose 6-phosphate, plus ADP, and that would be good enough for me, but you also end up liberating a proton. And so the substrate is glucose. That's your starting material. Question 26. Into what final product is glucose converted at the end of glycolysis? I'll just throw this out there to my students. Can anybody answer this one? At the end of glycolysis, not the citric acid cycle, but what's my glucose converted into at the end of glycolysis? Exactly, right? Because glucose has a total of 6 carbons, doesn't it? The formula of glucose is C6H12O6. But when we looked at steps 1 through 10 of glycolysis, we saw that somewhere around step 4 and 5, or step 4, that the glucose got split into 2, right? We end up making 2 molecules. And so we end up with 2 3-carbon units. And so the molecule that we end up with at the end of glycolysis is pyruvate, the structure of which is we have CH3, a carbonyl, then we have a carboxylate like this. And pyruvate is nothing more than the conjugate base of pyruvic acid, which is, if you just take this molecule, I'll just show you, if you copy it and you paste it, and all you have to do is remove the negative charge and put a hydrogen there, and you have pyruvic acid, okay? And you guys should remember acids and their conjugate bases. That would have been covered in Chemistry 101. So again, pyruvate is just the conjugate base of pyruvic acid. Okay, question number 27. Which statements concerning gluconeogenesis is false? Well, we learned that gluconeogenesis was the synthesis of new glucose from non-carbohydrate starting materials. A says gluconeogenesis is a pathway that is utilized when vigorous activity depletes our blood sugar and glycogen stores. Absolutely, we would use it then. Gluconeogenesis is the pathway, is the reverse of glycolysis. That is in fact false. And we went over that in lecture, and we said how steps 1 through 10 for glycolysis, those steps cannot be simply reversed in order to synthesize new glucose from non-carbohydrate starting materials. If gluconeogenesis was simply just the reverse of glycolysis, the 2 processes would be useless, right? They'd just be competing with each other. And so we looked at how steps 1, 3, and 10 of glycolysis are the irreversible steps. And so we use completely different enzymes in those steps during gluconeogenesis. So gluconeogenesis is not exactly the reverse of glycolysis. Yes, most of the enzymes are the same, right? Some of the steps are reversible, but steps 1, 3, and 10 are not. And so it is simply not the reverse of glycolysis. Question 28. Which of the following is a true statement concerning breaking a phosphoanhydride bond of ATP to form ADP? So if we crack that powerful phosphoanhydride bond, it says hundreds of kilojoules per mole of energy are released to form heat. Well, that's not true, because we end up forming how much energy? When we take ATP and we break it to form ADP, or we break that phosphoanhydride bond, we make the inorganic phosphate group, plus we release 7 kilocalories of energy per mole. So we actually only make, 7 kilocalories per mole, not hundreds of kilojoules per mole. What's next? Water is formed because the breaking of the bond is a hydrolysis process. No, we don't form water. It is an endothermic reaction. No, it's exothermic reaction because we release heat. Okay, so heat is released, therefore exothermic. Energy is released, which can drive biological processes like the phosphorylation of glucose. Absolutely. And energy is stored. No, we're actually costing energy when we use ATP to form ADP and release energy. Question 29. Which statement best describes how the interconversion of ATP and ADP is responsible for storing and providing energy for cellular reactions? Well, we've already looked at this, really. It says possible answers are A, energy is stored in ADP and released when ADP adds a phosphate group. That's just plain wrong. Energy requiring reactions of the cell drive the phosphorylation of ADP. No. Energy is stored in ATP and released when ADP undergoes phosphorylation. Also not true. Energy, however, is stored in ATP and released when the phosphoanhydride bond of ATP is broken to form ADP, and that is 100% correct. And then the last one, energy stored in ADP is released when the phosphoanhydride bond of ATP is broken. That is just plain wrong as well.